Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the three NIH-funded cryo-electron microscopy centers. My name is Christina Zemani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. Also with us today are staff members from each of the centers, including my colleagues here at NCCAT, Eugene Chua and Ed Eng. From the Pacific Northwest Cryo-EM Center is Marcia Mileto, and from the Stanford SLAC Cryo-EM Center is Nathan Burrows. For our main event today, NCCAT has invited our speaker, Biswanath Shah, who's joining us from Cornell University. And Eugene will introduce him properly after we get a quick update from each of the centers on the resources we make available at no cost to the research community. This webinar is a regular event we host the last Thursday of each month, highlighting users of the National Cryo-EM Centers who generously share their tips and tricks that have enabled them to make use of cryo-EM in their research. You can save the date already for our talk in June. June 25th, Professor Mark Herzig from the University of California, San Diego will be presenting Old Tools, New Tricks, Engineering Better Cryo-EM Sample Preparation Workflows through Design and Chemistry. I know our group here at NCCAT has been intrigued by some of the Herzig Group's recent results, so we're very much looking forward to that talk. And if you want to join us, you can already find the registration link on the events page at cryoemcenters.org, where you can also find registration for all of our upcoming talks, as well as recordings from previous talks. I did also want to remind everyone summer conference season is quickly approaching. There are a couple of meetings where you'll find center staff either presenting or attending. I wanted to highlight a couple of the bigger ones, the NISBRE meeting in a couple of weeks in Bethesda, as well as M&M in Milwaukee in August, and the IUCR meeting in Calgary. So if you wanted to find us in person, those are great opportunities. For final logistics today, if you have questions about accessing National Center resources, please feel free to use the chat to reach out directly to any of the panelists here. For questions for our speaker during the talk, please use the Q&A box. You can send questions at any point, but we'll hold them to the end, and I'll be moderating Q&A after the talk. So let's go ahead and get started. First, I will turn over to Nathan to let us know what's new at S2C2.
Hi, everyone. At S2C2, we have similar resources as the other national centers for high-resolution data collection, exploratory screening and optimization, as well as training. Today, I'd like to highlight that we are accepting applications for our summer workshop on SPA specimen preparation. It's a hybrid workshop from July 13th to the 17th for facility users who are interested in improving their sample preparation workflows. It's designed for researchers with prior cryo-EM experience, and this program supports those looking to adopt best practices and troubleshoot persistent challenges in their grid prep. The workshop will include both lectures and practical sessions. Lectures will be broadcasted via Zoom and will cover pre-sample screening, grid evaluation, grid selection, freezing instruments, strategies for mitigating air-water interface, as well as emerging vitrification approaches to improve your particle distribution and orientation. There are limited space available for the hands-on interactive in-person component to the workshop. The deadline for registration is June 12th, and selected participants will be notified by June 30th. More details on our website under the training section.
Thank you, Nathan. Now I'll turn over to Marcia.
Good morning, everyone, and my name is Marcia, and I'm a microscopist at the Pacific Northwest Center for Cryo-EM in Portland. And this is especially for those of you that are joining the seminar series for the first time. At PNCC, we have two types of proposals: a screening access that allows you to come on site for up to 5 days. And this is mainly aimed for like low resources institutions. We also have general access proposals, which awards researcher for up to 480 hours of microscope time of a year for 2 years. And this can be used both for screening and data collection. This last type of proposal are also eligible for participating in our training programs. And we have a variety of workshops that cover the entire Cryo-EM pipeline. And we just finished a very successful EPU data collection workshop, but there are more to come in the next months. And all approved proposals at PNCC are assigned a microscopist as a scientific point of contact. And this person will help you with questions about sample prep optimization, data collection, and so on, any question you might have about cryo-EM. We have 6 microscopes, cryo microscopes, Antarctica, Glacius 2, and 4 Cryos. Our 4 Cryos are equipped with Falcon 4i and Selectrix X and K3 and Biocontinuum in different configurations. We have Antarctica with Falcon 4i and K3 and a Glacius with Falcon 4i and Selectrix. And we have also a lot of instruments for sample preparation in case you're interested in that as well. And if you're interested in working with us, please go to pncc.labworks.org and apply. Our deadline for proposal review is the first of every month, and you will find all information on our website. But please reach out with any questions. And yeah, we're looking forward to hearing from you. So apply.
Thank you. Next, I will turn over to Ed to let us know what's going on at NCCAT.
Hi, I'm Ed, coming here from New York at NCCAT. Similar to the other centers, we offer training, grid preparation, screening, and data collection. We have quarterly calls for proposals. Our next call for proposals is July 1st. As Christina mentioned, it is summer conference season, so national meetings are very important, as well as regional meetings. Yesterday, we had the New York area Cryo meeting. Next month, we'll have the New York Structural Biology Discussion Group on June 11th at the CUNY Grad Center. And I'm also excited to invite you to the second annual Western New York Structural Biology Collaborative, WNYSBC Symposium. That'll take place July 28th at University of Rochester Medical Center. So if you're interested, the application deadline is July 7th. If you want to submit abstracts, that's actually going to be June 23rd deadline. So the event again is July 28th at the Rochester Medical Center. If you'd like to find out more and all about our training, screening, and data collection processes, you can go to ncat.nysbc.org.
Thank you, Ed. And now, Eugene, you can take the honor to introduce our speaker.
Thank you, Christina. Hi, everybody, and welcome to our webinar series. It's my pleasure today to introduce our speaker, Biswanath. He is a graduate student at the Department of Biomedical and Translational Sciences at Cornell University, working in the lab of Rick Sarion. And there, Biswanath uses genetics, biochemistry, and structural biology tools to understand the molecular mechanisms of cell survival. Prior to this, he did his undergraduate and master's research at the Indian Institute of Science, where he also has developed skills in X-ray crystallography, single molecule assays, and fluorescence microscopy. I had the pleasure of working with Biswanath last year through NCCAT's screening program, and I learned a lot from him about being meticulous and detail-oriented in doing science. So I'm really looking forward to learning more from him today. So Biswanath, can you please take us away?
Thank you, Eugene, for the kind introduction, and thank you, Christina, for the invitation. I'll share my screen. Hello, everyone. I'm Biswanath. I'm a third-year biophysics graduate student in Sirion Lab. in the Department of Chemistry and Chemical Biology and in the Department of Biomedical and Translational Sciences here at Cornell University. And today I'll be discussing about Cryo-EM sample preparation optimization. I'll use examples of my proteins, which I've been working with, the Dock7 and Cdc42, as an example to discuss about the challenges I faced while doing grade preparation and the strategies I used to overcome those challenges. Before we take a closer look at the results and the methods, let me give you some brief biological context of this study. So our lab has been looking at both of this protein, Dock7 and Cdc42, in the context of mTOR signaling and cancer progression. So mTOR is a serine threonine kinase. It is a protein. It is a sensor inside our cell which senses growth factors, nutrients, energy availability, and makes a decision for the cells if it has to grow, divide, or conserve its resources. And it does so by forming two distinct canonical complexes known till now, the mTOR complex 1, also known as mTOR C1, and mTOR complex 2, also known as mTOR C2. So the mTOR complex 1, so both of these complexes have some conserved components such as mTOR and Raptor, mTOR and MLST8. And in the case of mTOR C1, the Raptor is the defining component for the mTOR C1, while in the case of mTOR C2, Richter is the defining component. So mTOR C1 basically senses growth factors, amino acids, stress, energy levels, and regulates cell growth and proliferation, while mTOR C2 senses stress and regulates cytoskeleton dynamics, cell proliferation, and survival. Both of these complexes have been implicated in cancer progression and are known to promote oncogenesis. Our lab has been looking at this protein known as Dock7, and Dock7 is known to interact with a number of components of the mTOR signaling pathway. Dock7 canonically behaves as a GEF. GEF stands for guanine nucleotide exchange factor. It is an activator for GTPases such as Cdc42 and RAC1. It basically switches the GTPase from an off state where it is bound to GDP to an on state where it's bound to GTP. And the Dock7 has two conserved domains, the DHR1, or known as Dock Homology Region 1, and the DHR2 domain. The DHR1 domain has a C2-like motif through which it can interact with the membrane, while the DHR2 domain has the GEF site responsible for the GEF activity, the dimerization domain through which it can interact with another molecule of Dock7, and an allosteric site. So interestingly, Dock7 can act as an activator for Cdc42, and the activated Cdc42 can come and bind to Dock7 and have some effect on Dock7. So Dock7 acts both as an activator and an effector of Cdc42. So here is the AlphaFold predicted structure of the Dock7. Dock7 is a huge protein, and these are the DHR1 and the DHR2 domain of Dock7. So Dock7 expression levels are very high in triple negative breast cancer cells. And triple negative breast cancer is a aggressive form of breast cancer, which is very difficult to treat. And Dock7 levels are higher than the other members of Dock8 and Dock6, Dock3 subfamily. And we also did some Western blotting which showed that there's high levels of expression of Dock7 in triple negative breast cancer cells. So our lab discovered a new mTOR complex known as the DOCTOR complex. And this complex is distinct from the other known mTOR complexes that it is active in the absence of growth factors, and it helps cancer cells survive nutrient-stressed conditions. It has mTOR C1-like activity, but it is Raptor-independent, non-lysosomal in localization, and it is Cdc42 and Dock7 dependent. So here is how the mTOR complex looks like. We have the Dock7 represented here through the DHR2 and the DHR1 domain, AKT, TSC2, TSC1, mTOR. So whenever there's some sort of nutrient stress, the activated CDC42 comes and binds to the DHR2 domain of Dock7, and this leads to some sort of activation. And meanwhile, the DHR1 domain of Dock7 protects the phosphorylation of AKT from the phosphatases, which are activated during the growth factor deprivation. And this is a key signaling event which leads to the phosphorylation of TSC2, which gets inactivated, leading to the accumulation of activated Reb, which then stimulates mTOR activity, leading to S6K phosphorylation, leading to the survival of cancer cells. So one of the goals of my research is to provide structural information about this new mTOR complex we discovered. But as you could see, there are multiple proteins which are part of the complex. So initially, from a PhD, I started with understanding the interaction between these two key proteins, the activated CDC42 and the DHR2 domain of Dock7. So the other protein of interest in our complex is the CDC42. It is a Rho GTPase. And so it is one of the proteins which has been extensively characterized by our lab. It has three key regions: the switch 1 region, the switch 2 region, and the P loop. The switch 1 and the switch 2 regions are conformationally sensitive. So as you might recall, that CDC42 bound to GTP, or the activated form, was the one which was part of the Doctor complex. And so what is the structural difference between the activated versus the inactive form? So here's a comparison of the crystal structure of CDC42 in activated state bound to GMP-PNP, which is a non-hydrolyzable analog of GTP, compared with CDC42 in inactive state bound to GDP. And as you could see, there's most amount of changes in the switch 1 and the switch 2 region. So based on what we know, we think that the active CDC42 binds to an allosteric site, and this allosteric site is distinct from the GEF site where the inactive CDC42 gets activated or the nucleotide exchange happens. This hypothesis is based on the experiments done by former graduate students in the lab, where they were able to pull down using activated CDC42 full in Dock7, and activated 42. So Q61L mutant is a constitutively active version of CDC42, and it could stimulate S6K phosphorylation, which is a readout for mTOR activity. So CDC42 can stimulate S6K phosphorylation similar to the known activators of mTOR, Reb, and Hergelin in serum starved condition. So that's how we know that CDC42 is important for mTOR signaling. So the goal of my project was to first start by characterizing the interaction of CDC42 in activated state bound to the DHR2 domain of Dock7. And this would serve as like first step to understanding the whole Doctor complex. So we wanted to know through the structure that how CDC42 is binding to the DHR2 domain of Dock7. What was the stoichiometry of the interaction? What happens due to the binding of CDC42 when it binds to the Dock7? So what I did next was do some AlphaFold 3 prediction because one of the advantages of using AlphaFold 3 is that we could select a ligand of interest, such as GTP, and we wanted CDC42 to be in activated state to see how or where the CDC42 binds to the DHR2 domain of Dock7. So this is the structure. This is the prediction. And the prediction is pretty confident. So in cyan and green, we have the DHR2 dimer. So this is one of the unit, and this is the other unit. While in yellow and magenta, we have the CDC42 in activated state. And what AlphaFold 3 predicts that CDC42 is bound to the GEF site. But based on the experiments done by former graduate students in the lab, biochemical experiments, we were thinking the allosteric site is the one which is involved in this interaction. But AlphaFold 3 prediction suggests that it wasn't. So we decided on determining a cryo-EM structure of the complex. So we started by purifying each of the individual proteins from expressing and purifying it from bacterial cells. So we use CDC42 having a Q61L mutation. This mutation makes the protein constitutively active by making it GTPase defective. So it is always bound to GTP. This is a site exclusion chromatogram and the corresponding SDSP gel. Again, for Dock7, the full and Dock7 protein has been difficult to express and purify. And so we stuck to the DHR2 domain because we know that the activated CDC42 bound to the DHR2 domain of Dock7. And we used a slightly truncated construct of DHR2, which had high levels of expression and still had both the allosteric site and the GEF site. So we used sumotax for better expression of the protein. The protein was not very stable, and we found out through trials and error that it was only stable at high salt concentration greater than 300 millimoles and high glycerol greater than 5% volume by volume. So this is the site exclusion chromatogram and the corresponding SDS-based gel for the protein. So now we have purified both the proteins, and next we wanted to isolate the complex. So I incubated 4 times molar excess of CDC42 with the DHR2 domain of Dock7 overnight and passed it through the sizing column. And so here is CDC42 co-eluting with the DHR2 domain of Dock7. And so now we were able to isolate the complex. Next, we wanted to move on to the structural studies. So we started by looking at the complex under negative stain electron microscopy. What we observed, so this is like, so our complex was plotted onto the negative stain grids using a uranyl acetate stain. And what we observed, there were a lot of single particles, but in the background, there was a lot of aggregates too. And I thought that the aggregates was there because the uranyl acetate stain is has a very low pH. And I knew that the protein has a tendency to aggregate and precipitate out at very low pH, causing those aggregates. And also the resolution we could achieve using negative stain electron microscopy is not very high. So we decided to move on to cryo-electron microscopy. So this was some of the initial grid screening we did here at Cornell and on the Arctica. And so I plotted 3 mg per ml of complex onto the cryo-EM grid. And what I observed that most of the protein formed aggregates and there were not single particles visible. And only when I reduced the concentration of the particles to like a very low levels, I could see some single particles in the micrograph. So what I did was collect a data set on this cryo-EM grid and looked to see how the 2D class averages looked like. So this is what the 2D class averages looked like. As you could see, it looks like a bean-shaped structure with no details and very blurry. And this is what the 3D reconstruction looked like. So which did not look great. And I tried a few different conditions and things were not working out well. So I needed some expert help. And that's when I decided to apply to the NCCAT GUP3 proposal. And I was delighted when it got accepted. So we traveled to the New York Structural Biology Center. And here is me talking with Eugene. And here is Sean. Sean is a research associate in our lab, and he's expert in crystallography. So when we reached the NCCAT, the first thing we did was do some concentration gradient to see how the particles behaved at different concentration. And at high concentration, what we observed was that the particles formed a lot of aggregates. There were no single particles visible. And as we reduced the concentration, the aggregates started to reduce, and probably you could see some single particles. And at a very low concentration, there was very less aggregates. However, the single particles were also very less. So we wanted to know if the protein we plotted onto the cryo-EM grid was already aggregated before we put it on, or is the aggregation happening during the process of grid preparation? So we use an instrument called mass photometer available at NCCAT. So what mass photometer does, it basically measures scattering of light from a particle. So what we do is we add some sample onto the glass slide and see how the light is scattered. And based on how the light is scattered, the mass photometer gives you a histogram of different molecular weight species. And so that's what we did for our sample. And this is the histogram we obtained from the mass when we put our sample for the complex onto the mass photometer. We observed uh, three three peaks, one corresponding to 104 kilodalton, 236 kilodalton, and 447 kilodalton. So we knew that DHR2S is known to form a dimer, so we're expecting a peak around 104 kilodalton or higher, slightly higher for if it binds to CDC42. But we did not expect these higher order oligomeric peaks, probably corresponding to a tetramer and an octamer. So that was something interesting we got to know. But however, we did not see like aggregates when we were looking at under mass photometer. So we thought that the aggregation is happening during the process of grid preparation because of a problem known as the air-water interface. So ideally, when we blot our sample onto the cryo-EM grid and vitrify it, we would expect that the sample to be well distributed, uniformly distributed in the thin film of ice. But sometimes what happens that's some of the samples try uh, uh, some of the samples prefer the air-water interface because it is hydrophobic, and the hydrophobic patches in the sample tend to stick on this hydrophobic surface, leading to sometimes preferred orientation, a dissociation of complex, and probably in our case, protein denaturation. So one of the ways one can overcome the problems of air-water interface is to use detergents. And what would detergents do is stabilize those hydrophobic patches and prevent the protein from going to the air-water interface. So we screened some of the detergents and found that CTAB at 0.1x CMC and Chapso at 0.1x CMC seems to help with the dispersity of the particles. Uh, it so basically it helps stabilize the dimeric form of the protein. And in case of Chapso, it increased the dimeric form to very high levels. So now we wanted to see how this addition of detergents looked under the microscope. And this is how the micrograph looked when we substituted CTAB at 0.1x CMC. As you could see, there were far less aggregation. We could even see some single particles. But for Chapso, we weren't able to see even particles on the micrograph. And that is because when we are trying to use detergents, we want to generally have very high protein concentration. And because proteins tend to stick to the carbon layer. And so another way to overcome the air-water interface problem is to use specialized grids known as the graphene oxide grids. So graphene oxide is a highly oxidized form of graphene, and it has functional groups which are projecting out of the surface. So we put a layer of graphene oxide on cryo-EM grid, and then we blot our sample onto it. So this is how the micrograph looked. And so we collected a small data set on this micrograph and to see how the 2D class averages looked like. And since the graphene oxide coverage wasn't that great, we got some few particles which looked like this, and they vaguely resembled the AlphaFold predicted structure of the DHR2 tetramer. So what we have been observing here was mostly tetrameric form of the DHR2s. So this was some of the experiments we did in the first GUP3 session at NCCAT. We weren't able to figure out good cryo-EM conditions to determine the structure of the protein and get a good distribution of particles. So Eugene suggested that I do some biochemical optimization and look at the data we got in the first session, and then come back to the NCCAT for another GUP3 session. And so I went back to Cornell, worked on some biochemical optimization. The first thing I did was switch columns of size exclusion chromatography from SuperDex 75 to SuperDex 200 for better resolution of higher molecular weight protein. And indeed, I saw that there were a lot of higher order oligomers. And so I wanted to minimize those higher order oligomers, and because we were only interested in the dimeric form of the protein. So as you might recall, that CHAPSO helped with increasing the proportion of the dimeric form and reducing the higher order oligomeric form of DHR2S. So I tried replicating those experiment using size exclusion chromatography. However, when I added CHAPSO at 0.1x CMC and 0.5x CMC, I wasn't able to see any difference. And even at higher concentration, CHAPSO seemed to have increased the formation of the multimeric species. So CHAPSO didn't seem to work out. So I did some screening with other additives and found out TSEP, which is a reducing agent, seemed to help with the dispersity of the protein. So when I added TSEP and made the environment reducing for the protein, I was able to get the dimeric form of the protein more, and I was able to get rid of the higher order oligomers. And next, what I did was repeat the same experiment with DHR2S, but along with the activated Cdc42 to see if I was able to form complex even in the presence of TSEP. And yes, I was able to isolate the complex. So the next thing we wanted to do now to see is how those particles looked under the electron microscope, or how the particle distribution looked in the micrograph. So again, I contacted Eugene and then returned back for another GUP3 session. So I took the bus from Cornell University to New York City. And yeah, so we looked at different concentration of DHR2 under the electron microscope to see how the micrographs looked. And as you could see, in the presence of TSEP, there is very minimal aggregation. And we could see some single particles. But as you see, the particles are the micrograph is very dense, is very busy, and the particle size is smaller than what we were expecting. And reducing the concentration by twofold did not help with this. We didn't see different orientation, or even the size of the particles seemed very smaller. And so all of these experiments were being done on quantified holy carbon film. So we thought of switching to a different type of grid, such as gold foil and graphene oxide grids. So with gold foil grids, we were able, the particles behaved slightly better. We were able to see slight more orientation. The particle appeared, seemed less busy. And in the graphene oxide grids, we're also able to see some more orientation, but still the particle distribution did not look good enough for data collection. And the particle sizes were still very small. than what we would normally expect. So we thought that this could be a problem with a problem posed by the air-water interface, and maybe the particle is having a preferred orientation because of the interface. So we added detergents. So as you might recall from the previous experiments done using mass photometry, Chapso seemed to help stabilize the protein. And so we tried our Chapso at 0.1x CMC, and this time we had like a high concentration of the complex. And this is how the micrograph turned out. As you could see, the particle distribution looks much better, and the particles are the size we would expect, and we could see a multiple different orientation. And this is some of the 2D class averages we obtained from some 50 screening micrographs. So next, what we decided was to do a data collection, and we used a GOP1 proposal to collect data on these grids. And this is how the 2D class averages turned out from the data collection. And I was able to determine a 2.74 angstrom structure of the complex. So this is a cryo-EM density for the complex. In purple and magenta, we have the DHR2 domain of Dock7. And in green, we have the CDC42 having the Q61N mutation bound to the allosteric site. And as we had predicted that or hypothesized that the allosteric site is sort of proximal to the dimerization site, and this is what we got from the structure. And this is another orientation showing the density for GTP, suggesting that the CDC42 was in activated state. Another orientation, and here's a local resolution estimate for the Cryo-EM density. So what helped me with the data processing workflow was the manual available through the NCCAT, the SPA processing manual for CryoSpark. I almost followed this manual, and the tutorials available on YouTube by CryoSpark helped me with the data processing. So I followed this workflow where I imported movies, did patch motion correction, patch CTF, curating exposure, doing a blob picker, inspecting pics, extracting them, and doing an ab initio and heterogeneous refinement. So I repeated ab initio, heterogeneous refinement, and non-uniform refinement till I get a good subset of particles, and through which I was able to get like a very high resolution structure. What also came useful was the NCCAT office hours. They provided me with nice suggestions on how to improve resolution, on how to refine the structures better. So yeah, so next what I did was fit an atomic model to the Cryo-EM density. And this is the atomic model of the complex. So in pink and purple, we have the DHR2 domain of Dock7. And CDC42, which is bound to the allosteric site, which is proximal to the dimerization site. And this is another orientation. So the same thing. So we took a closer look at the interface of the structure, and we found out that the residues which were mediating this interaction was the switch to region of the CDC42. As you might recall, switch to region is conformationally sensitive. And so when the CDC42 is in activated state, the switch to region protrudes slightly more, and it sort of docks into the DHR2 domain of Dock7. And these are the residues which I predicted are important for this interaction from using PDB-EPISA. And this interaction has been seen before, especially in the case of RAS, activated RAS and the RAS-GEF son of 7-less, which is a classic example of the allosteric interaction. And that interaction was also mediated through the switch to region. I also looked at how conserved are these residues in the doxy subfamily of protein. So the residues which were mediating the interaction with CDC42 at an allosteric site were not well conserved, except one residue, which suggested that Dock7 was forming a very unique interface for interacting with CDC42. So to conclude, some of the take-home points from the study was the best thing to do is to ensure that the protein sample which you're working with is highly pure, stable, and monodispersed. For my case, before grid preparation, it's very important to carefully screen the buffer composition, salt concentration, pH, and additives to improve particle behavior. And screening different types of grids and support films can improve particle distribution and orientation. For example, in my case, where gold foil and the graphene oxide grids showed better particle distribution. And detergents could also, well, especially in my case, where adding chaps help give us a better reduce the orientation bias and get a better distribution of the particles and avoid the problems posed due to the air-water interface. Yeah, thank you for listening to me. And I would like to thank my advisor, Rick, for his support and encouragement. I'm also grateful to Eugene. Eugene has a very helpful throughout this whole study of this complex. And I learned a great deal about cryo-EM techniques and methodology from Eugene. And as well as everyone at NCCAT who helped me troubleshoot grid preparation and data processing. Thank you. And I'm happy to take any questions.
Thank you, Biswanath. That was fantastic. And really nice. Like it's just so striking how much better those micrographs were.
Yeah.
When you see the final result, you appreciate how little you were seeing
Yeah.
in the earlier versions. That's so exciting. So our audience can send questions in through the Q&A box. We've got one already to get us started. But go ahead and send them in as we go along. So someone was asking what TSEP concentration you were using.
So I was using the normal TSEP concentration, like 1 millimolar, not really very high. I think the protein was very much sensitive to either oxidizing or reducing condition. And I think that made a difference.
Yeah. All right. And there's another question. Your salt and glycerol concentration in that final sample that made such a nice grid, were they as high as you initially said, or what were those final concentrations?
Yeah, I think the salt concentration, which was there in the buffer I used for getting the final structure, was around very high. It was like around 500 millimolar salt and 5% glycerol volume by volume. Yeah.
Impressive. We don't normally suggest that, but you're proof that it can be done.
Yeah. Unlike the protein was only stable at those concentration, and it would aggregate if I reduce the glycerol concentration or salt concentration.
I'm a little curious. This is a question from me, because I can.
Mhm.
Were there other additives you tried?
Oh, I tried so I did like a differential scanning fluorimetry, and we had like sort of, I would say some, I would say I tried glycerol. What else? Yeah, some detergents. So we had some screens. I don't recall the exact like composition of the screens, but we used some I think screens available through Hamptons for like screening conditions which made protein more stable. So I tried out those conditions to see if it helped. But most of them were just increasing like salt concentration or you adding like by like glutamic acid or arginine to make it more make the protein more stable. Uh, Yeah, mostly those.
Thanks. Were you using other reducing agents prior to adding TSEP as an additive?
I tried DTT before, but it didn't help that much. But I think TSEP made a lot more difference. I think because TSEP is slightly more stable compared to DTT. Yeah.
What was your approach to narrow down your detergent choice to Chapso?
Yeah. So when we were at NCCAT, we were doing this mass photometry experiments, and I think uh, we had like 4 or 5 detergents we were screening, and out of those detergents, I think Chapso and CTAB were seemed to help with the dispersity of the particles, and we thought that the dispersity of the particles the improvement of the dispersity of particle was because of it was increasing the stability of the particles, and and in case of Chapso, it improved the dispersity much more compared to CTAB. So that's why we decided to go with Chapso, and ultimately, Chapso seemed to help with the distribution of particles.
Thank you. There's a question that starts with a compliment. Very nice work. And this is more of a biology question. Do you have any insight on the implication of the CDC42 binding to the allosteric site? And do you see any population of it binding to the GEF site?
Oh, so yeah, answering the second question first, there is a small proportion of CDC42 which, even in activated state, does bind to the GEF site, like a very small proportion. Like transiently, the interaction is not very stable, and we are still looking at the biological uh, like the implications of the study. Right now, what I'm doing is mutating those residues and seeing how it affects the survival of cancer cells or even the metastasis in growth factor limited conditions.
Thank you. So there's a question about how your solve structure compares to the predicted AlphaFold structure.
Yeah, the predicted AlphaFold structure suggested that the CDC42 inactivated site would bind to the GEF domain, the GEF site where the GEF exchange is GEF activity is happening. But the determined structure suggests what we found was that the CDC42 is bound to the the allosteric site, which is sort of away from the GEF site, and the GEF site is sort of empty. And so it's sort of like very different, it and is more the alpha predicted like where CDC42 would just normally bind when it is like in active state. Yeah.
So actually your mass spectrometry results showed some higher molecular weight species. Did
Mhm.
you see any hints of that in your micrographs? Um, or were you only getting your dimer that you were expecting?
Yeah, I'm like, so I haven't like so what I observed that the isolated domain of the DHR2 has a tendency, uh, even in the presence of TSEP, to form like higher order oligomers, even filaments, but which we normally don't see in cells. I think it is more like of an artifact. And yeah, like if we go beyond a certain concentration, we have observed that it forms, even in the presence of TSEP, it forms this tetramers. But we're not sure because we haven't seen any filaments or similar structure when we have been doing some microscopy experiments. So I don't think the full-length Dock7 would actually form filament. It is because we are working with an isolated domain. It has a sort of tendency to form those filament or like higher order structures.
Thanks. Um, Did you try different freezing, sort of plunge freezing conditions? Did you need to optimize things like blot time and blot parameters?
Yeah, I think one of the advantages of working at NCCAT was that we already knew like what were the good blot times, and for example, like for some grid conditions, we had to optimize. For example, graphene oxide grids, we had to optimize the plotting conditions, but for most of the part, for the gold grids and the holy carbon grids, we used like 3 second, I think 2.5 or 3 second plotting time, and I think zero blot force. Yeah.
Thank you. Back to another biology question. There are several human genetic mutations in CDC42 leading to developmental and immune syndromes. What's the consequence of these mutations with regards to your CDC42 Dock7 complex?
I'm familiar with few mutations, but I'm not so much familiar with all the genetic mutations. Impo- so I have to look it up, but yeah, I'm not really sure if
Yeah, we can.
if this yeah, I'm not really sure about that.
All right. I think we've gotten to all of the questions that came in. Um, so unless any pop up in the next couple seconds, we can go ahead and wrap up. Thank you everyone for joining us today. We wish you all as much luck in your cryo-EM endeavors, and the national centers are here to help support your science. So let us know how we can help, and we hope you see you again at one of our upcoming talks.